Hello. If you're viewing this presentation, it's likely because you have been referred to Travera and you're interested to learn more about our rapid cancer therapy guidance test and whether or not it might apply to you. So it's first to understand what our goal is as an organization. And here at Travera, we're trying to help increase the likelihood of a clinical response when patients move beyond standard of care treatment options in advanced disease or in rare disease. The reality is in the US, most physicians follow the NCCN guidelines, which is a phenomenal tool that's based on published data, peer reviewed aspects, et cetera, to help guide decision making in cancer therapy and cancer therapy guidance. However, when you get into late stage disease and stage four and metastatic disease, what you quickly find is that guidance becomes less directive. And if you've gone through a first or second line of therapy, you might find yourself in a position where you're now looking at a long list of potential drugs, potential regimens, and the third line sequence of drug selection is not really clear. And so your physician uses a lot of different resources to decide where they're going to go next. They might use genetic or genomic testing. They're going to look at what you've done well with or tolerated well in the past. They're going to have a lot of different conversations with you and analyze a lot of different aspects of your clinical situation to help make this decision. But with so many options in front of them, the reality is that there's a little bit of educated guesswork that occurs. And what happens is that you try a regimen, you try a new treatment, in your third or your fourth line of therapy, and you wait to see if it works. And if it does, great, you continue it. If it doesn't, then it's now time to switch to something different or something new. And so it becomes a little bit of a game of cat and mouse. And so what our hope here at Travera is that we can help enhance this decision-making process with informed information on what your cells are responding to in a very rapid manner. And we do this by kind of stealing from a theory that's been used in infectious disease for a really long time. There's a tool called an antimicrobial susceptibility test, or an AST. And if you had an unusual infection and you go to see an infectious disease specialist, they're very likely going to take a small biopsy of your infection, and they're going to grow it up in a petri dish. They're going to test a variety of different antibiotics on it, find out what worked and what killed those cells, and they're going to come back and that's what they're going to give you. This is a highly successful approach. Why wouldn't it be? It's very logical. So why not do this in cancer? It's a great question. And in fact, we have for a really long time. In fact, this has been tried since the 1950s in oncologic medicine to really just duplicate the success we've seen in infectious disease and test drugs ex vivo, or ex vivo drug testing, or out of the body drug testing. Unfortunately, in 70 plus years of research and clinically available tests, there have been numerous failures and just really inconsistent success in this space. And so it's important to understand what has happened so you can understand what Travera is doing differently. First of all, you have to start with live cancer cells with these tests. It's really difficult to determine if a drug's going to kill a cell if that cell's already dead. So you need a fresh biopsy. You need a fresh sample. And so you take those cells out of the body, and you're faced with your first real challenge, which is when a cancer cell is removed from the body, it typically dies within about 2 or 3 days. And so in that time, you're facing the clock and that natural cell death. The second real challenge here is that drugs take about 5 days to kill the cell. So it's tough to measure the cell death based on a drug when the drug takes 5 days to work, but the cell dies by itself in 2 days. So what have we done? We've intervened. And we've intervened in a lot of different ways. First of all, we tried exactly what they do in infectious disease. Put it in a culture, put it in a petri dish. Let's grow the cells and let's keep them alive long enough to test the drug. We've moved on to trying that in mice and duplicating tumor growth in a mouse that is genetically identical to what is in your body. And let's see what drugs work in these mouse models. And more recently, there's a technology called organoids. And an organoid is essentially that petri dish culture, only instead of being grown flat across that petri dish medium, it's grown in a three-dimensional way, which in theory maintains more of the microenvironment of the individual cell, of the individual tumor. The challenge with all of these approaches is that they take weeks, in many cases, sometimes even longer than that. And we measure drug efficacy, but the difference between cancer cells and bacteria in the infectious disease example is that Biology in cancer is different. Cancer cells aren't bacteria. Cancer cells are mutants and they're mutated cells. And when we intervene in such a significant way, the biology of cancer cells inevitably change. And so what's tested against drug here doesn't necessarily equal what was pulled out of your body there. And so when you take this concept to your doctor, they may have experience with these previous generation tests, and their response might be, hey, I appreciate the suggestion, but ex vivo drug tests just don't work in cancer. And they're not wrong. But what has changed is the addition of a new measurement tool. And measurement tools change science. Think about the invention of the microscope or of the X-ray machine or of an MRI. All of these measurement tools changed science and changed medicine. And so there was a new measurement tool called the suspended microchannel resonator developed at MIT in a biophysics lab there. And essentially what this does is this very small chip you see on this image here. And what we do is we take individual cells and we pump them through these little blue channels very quickly. And as we do, they pass over this little cantilever beam that looks kind of like a diving board. And just like a diving board, when you jump off of it, it vibrates. And the vibration of that diving board is different for a full grown adult versus a young child. And so what we actually measure is the difference in that vibration, that frequency change that occurs. And it's such a precise level of measurement that we're able to see the weight of an individual cell that's 100 times more precise than any other tool that exists today. So the team at MIT took this to the team at Dana-Farber Cancer Institute, and they said, we know that it takes about a week for a cancer drug to kill a cell, and we know when the cell dies, it loses about half of its weight. So our question is, if we apply the drug to the cell, might it lose a small percentage of its weight in the first day, maybe 3 or 4%? And the answer to the question, after years of research, was yes, it does change that much. And we're able to measure it. And so the way that this assay actually works is we get a sample of live cancer cells. We would put 5,000 cancer cells in this gray cup and 5,000 cancer cells in the blue cup. We apply drug to the cells in the blue cup, and we don't do anything to the cells in the gray. After 24 hours, we go back and we measure every single cell. All 5,000 cells in each cup are measured individually. And from those individual measurements, we build these little graphs. If the graph of the control sample in the gray looks exactly the same as the graph in the blue sample, and those two things overlap, it tells us that the controlled cells and the drug cells are changing at the same exact rate, which means the drug's not doing anything. It's basically a placebo. It's not working. But if those two graphs look statistically different, it tells us that the drug is having an impact on these cells, and enough impact that we know the drug is working and it's doing something different as these cells move towards death 4 or 5 days from now. And so this is the first ex vivo drug testing in the history of 70 years that actually tests drug response against your cells and not a grown version of those cells. So how does this translate to outcomes and individual patients from the lab? And the question is so far very well. The science is early and the data is young and the numbers of patients are small. But we've done this test in a couple hundred patients. We have outcomes so far on just under 50. And what this means is these are patients that received the test, received one of the drugs that were tested, and then we waited a month or two to see whether or not the patient responded to that drug. And right now, the predictive accuracy in these 49 patients is 84%. That means 41 out of 49 patients we predicted correctly whether or not they were going to respond to the drug that they received. So if you're on your third or your fourth line of therapy and you're looking to switch to the fifth, imagine the ability to look at that next list of drugs and analyze your cellular response to 20 different drugs in our test, in our hands, in our lab. And in 2 days, getting a report back that says, here's 20 drugs, and here's how your cells responded to each, so that you can make a better informed decision on what drug you're going to choose next with about 84% accuracy. This is what we're working towards. This is what we're doing now. And as we grow the number of patients in both clinical trials and in early access, we expect this data to maintain this high predictive accuracy. And we're excited about the opportunity to work with you in doing so. We have this available a couple different ways. Our early access test is likely how you found us. And right now, we're not billing anyone for this, not your insurance, not Medicare, not you. We're willing to do the test for free with the understanding that this data is young, that we are still learning, and that we want to learn together. Some of these tests will go beautifully. Others will learn things through, and they may not work, meaning we may not be able to produce a result for one reason or another. Our only real ask in this approach is that we're able to find out how things went. What was your experience with the test? What was your doctor's experience with the test? And did you use one of the drugs that we had in our panel? And if so, did you respond to it positively or not? And we collect all of that information through a really simple online HIPAA survey that takes about 10 minutes or so to complete, and that we'll send to you, a month or two after you've received your treatment. The other option is to do this through a clinical trial, and we have several clinical trials open. One is through a company called XCures, and what's nice about this trial is that it's an N of one trial format, meaning that your physician that currently is treating you in the facility you're currently attending doesn't have to be a trial site for you to participate. You can actually go directly to the XCures website and sign yourself up for the trial, and the XCures clinical team will reach out to your physician and coordinate the collection of the sample and the ongoing outcomes information that's needed as part of the trial design. It's a really nice way to participate in the clinical trial. It's very easy to do. So who qualifies for this? Quite simply, somebody that's planning to try a new therapy is one of the first requirements. If you're on a therapy today and it's working and you're responding to it, that's great news. You should keep doing what you're doing. However, if you're having increased toxicity, or if you just, experience progression and you're looking to change to a new therapy, that's a great time point in which to try our test, because it can help you to make the decision and your physician to make the decision on what to try next. We do need live cells, which means we need a fresh biopsy or fresh fluid collection from a pleural effusion or abdominal ascites. We can work with you on how to approach that process. But your physician is likely to order a fine needle aspiration or a cord needle biopsy on you for lots of different reasons. And generally, we will just partner through that process and take a portion of the sample that's being collected for other clinical needs. We cannot today do this on bone metastases or cerebral spinal fluid. It does have to be a soft tissue biopsy from either a metastatic lesion or a primary lesion. We are a CLIA approved test, which means we are approved at the state and federal levels through that CLIA approval, and that does mean that this requires a medical physician to order the test. We can work with you through that process. The only state where we are pending our approval, simply because their process is slightly different and takes a little longer, is New York. But there are exceptions that exist, and so if you're a New York resident, we can even work with you through that exception process. In short, if you're interested in pursuing this, all you need to do is contact us and let us know of your interest. You can go directly to the XCures website at www.xcures.com/advanced-carcinoma to learn more about the trial, or you can contact me directly. My name is Dennis Watson, and I'm the Vice President of Business Development here at Travera. That's dwatson@travera.com. Of course, reaching out is no commitment to do anything. We're happy to answer any questions you may have, outline the process and next steps, and even support you in communicating to your clinician what this test is and how it might benefit you. Thank you for your time. We certainly do appreciate it and hope to hear from you soon.